Okay, so biophysical models of single neurons. I'm going to really try to keep it fairly basic. This crowd, probably I don't have to talk a lot about why we want to do models, but I will still give you my opinion on that. I will then say what really is a model, and I'll introduce you to an overview over different types of models, and then I'll go into the particular type of model that Tim has kind of already motivated with the Hodgkin-Huxley model, and that is what's called conductance-based models. So we'll again talk about sodium and potassium, but also other conductances. And then I'll go a little bit into more recent work from the lab where, together with my former postdoc, Cengiz Günay, we constructed a single compartment model of a fly larval crawl motor neuron. And there are reasons for that I can get into, but I wanted to introduce that model because I have some exercises that we can do this afternoon that are using this particular model in a modeling software called XPP, which is really a differential equation solver. So for those of you who have not seen the actual nitty-gritty of a single compartment model neuron, we can actually look at the code. The reason I chose XPP is that it's fairly intuitive code, and then there will be an interface where you can play around with this model and change different parameters and kind of try to get a feel for how different conductances interact in a model with richer dynamics than you would get out of an integrated fire neuron. So why modeling? That gets me again into this question that Tim kind of talked about, and that is, do we really need all the complexity? And the Hodgkin-Huxley that he introduced isn't even all that complex. It just has a few voltage-dependent currents. What do we get out of that, or can we get away with simplified models like the Fitzhugh-Nagumo or the leaky integrated fire neuron? And I would argue that for this question and for basically any question where you think about modeling and what kind of model do you want to build, it really depends on the question, the scientific question that you want to ask. So if you're just interested in spike times, then we just learned that some of these very simplified models can do a very good job, but I'm going to show you some richer dynamics for which, if we want to replicate it and understand it, I think we actually need to model these conductances in detail. But the basic story is always, don't just launch into a model because your advisor says so, but think about what am I trying to achieve here? Do I need dendritic complexity? Can I get away with a single compartment? And that choice of model is one of the most important things if you want to do computational neuroscience. You can really run yourself into a corner if you pick the wrong kind of model. So that gets us back to this complexity. Just a reminder, you all know that we have in neurons that are connecting to each other synaptic conductances and membrane conductances. And we've heard a lot now about the generation of this action potential. And I would kind of liken this to what may be a little bit of a tacky analogy. I would say that we have ion channel types in a single neuron that you could compare to types of musical instruments. So they each have their own properties. You have violins, you have drums. All of them have slightly different properties. And so all the ion channels that are present in a neuron then form what's kind of like an orchestra. And I'm making that analogy because again, I want to emphasize the rich dynamics that you can get even at a single neuron level. And so what we're really interested in is the membrane potential that kind of corresponds to the music generated by this orchestra. Okay, was that too tacky of an analogy? No. And please interrupt me. I think toward the end of Tim's presentation, we had a nice discussion going, and I think for the first day of a summer course, that's a good thing, so let's keep that up. And I tend to kind of drone on and on, so I'll try to ask you questions, but also please interrupt me and make comments or ask questions. So why modeling? Why not just stick to experiments? If we're interested in how the brain works, we should be studying the brain, right? There's a quote from someone, I forget who, and I forget the exact wording of the quote, but I think the quote goes something like, the best model for a cat is a cat, right? So why shouldn't we learn what we can just from experiments? Why do we need models? Neurons and networks are highly nonlinear and complex systems, and you have already seen that even at a single neuron level with a single compartment where you're not even bothering to describe the whole morphology of the dendritic tree, you can get complexity that 4 and higher dimensional becomes hard to understand. So studying models can be useful in studying these nonlinear complex systems, because in a model you can really manipulate whatever you want, right? The equivalent of doing like a ion channel mutation in an animal that requires a lot of experimentation and a lot of preparation, you can just do by changing a couple of parameters in your model and, change the properties of that ion channel or get rid of it. So in that sense, models can save us some work. We also have many parameters and variables that are not, at least not currently, experimentally accessible or manipulatable. And so again, in a model, you have complete control over things that you could not change biologically. Like I could say, what if I make my membrane capacitance twice as big as in the original? You can't do that in a neuron. You can't suddenly make that neuron bigger, right, with a higher capacitance. So you have this complete control. You can vary parameters individually, which often, with dirty drugs that you try to block just one ion channel, but they interfere with other ion channels as well. In a model, you can change every single parameter individually, and that is often an important way to get into first insight into what do these different components of the neuron do. But you can also, and a lot of my work in recent years has been dealing with that, and people's work also. You can look at what happens if I vary different parameters individually at different ratios, and it turns out that biological neurons actually seem to be doing a lot with such what we call co-regulations, with kind of interrelationships between parameters. So you can do that in your model. I have been known to be an explorer of parameter spaces. So if I have time this afternoon, I might tell you a little bit about that. That's work that started mostly as a postdoc with Eve Marder, where we basically now have, even at the single neuron level, and I also study small networks, we have so many parameters that we're talking about a high-dimensional parameter space, and it's very counterintuitive to understand what's going on in these spaces. And so there are different ways to do this exploration. I have been doing a lot of what I call a brute force approach, where I just cover the entire high-dimensional parameter space with simulations and ask what's going on in different parts of that space. And as I said, I don't have that included in this talk, but if we have time and if there's interest, I could do like a half-hour thing on that this afternoon. You don't have to sacrifice animals, and the modeling, once you have a model that works, is less time-consuming than experimental work. And I say this from a perspective of someone who has done both, right? So, and what I also need to say from that perspective, I assume that most of you here are, who here is actually doing experiments with neural systems? It's about half, actually, good. So from that perspective, as someone who myself and in my lab is combining experiments and modeling, I want to really emphasize that we don't want to model in isolation, like in detachment from the actual biology. And I think Tim had a quote about that earlier, about you had a quote about how experiments are the gold standard, basically. And I want to emphasize that I really caution everybody not to do models in a vacuum. You always want to, either yourself or with a collaborator who you can talk to, and that's often difficult to find a common language. You should always try to keep things grounded, because otherwise you're mathematically exploring something that doesn't teach us anything about the brain. So that is my philosophy, and in some cases integrated fire can do that, in other cases it cannot do that. But again, it's always important to think about what am I trying to achieve here, and to have this close interaction between experiments and modeling. Also, it's not just us geezers telling you should ground your models in experiments, it's also the funding agencies. So if you have, and I think they have a point there, if you have...
I just got it. So, point in case, I just got a model. We just submitted a paper with just a model, and both reviewers said that's great, and the editor said, what's the biological implementation? And if you believe in MAR and you believe in 3 levels of modeling, then you're not always looking for a mechanistic implementation. So I think sometimes it's okay to go abstract as long as there is a biological question behind it. And I think it's on you guys and on us to educate the rest of neuroscience that it's okay for models not to showcase which ion channels are responsible for what. There's also a greater picture to be had in mind.
That's true, but it's both reviewers and journals and, as I said, funding agencies, and I have had multiple grants rejected where there were experiments and there was modeling, and then they're harping on it not being integrated enough, even though I constantly preach, you have to go back and forth. Right?
We have to do both, right? We have to survive in the current
funding
system, and we
climate,
also have to keep working on making the system into one that we think is more conducive.
Right, yeah.
Kind of thing.
So kind of work with the system as it is, but also try to change it. And I think that computation and modeling have over the last couple decades become more and more convincing to the rest of neuroscience. I think the times where people say, well, what is this model? That's really over. I think people respect the power of modeling, but you have to integrate it well with experiments. Okay. So what really is a model? And I don't have to say much about that because Tim set this up beautifully. To me, a model, at least the type of model that I'm working with, conductance-based models, are just a bunch of differential equations and parameters in a computer that you then solve numerically. So this is an overview over a model of the stomatogastric neuron in the lobster or crab. And this now should look totally familiar. You have a differential equation, and I just noticed there's a capacitance missing here. You have a differential equation that describes how the voltage changes based on the sum of all currents. And then here I'm saying times 8 because this particular model of the stomatogastric cell has 8 different membrane conductances. So a little bit more complex, but still just a single compartment, single neuron. And these now also should look familiar. You have m to the third h in the case of the sodium channel, or in general m to the p times h to the q. You have your driving force. You have these differential equations for the gating variables. So this is the basic structure of these conductance-based models. I have over here also a box for calcium dynamics, but we're not going to get into that today. But often in conductance-based models, intracellular calcium concentration is a very important variable that you then also have a differential equation for. Okay, so that is what I mean when I say model neuron. There are different other types of model neurons, and I'll show you an overview slide in a moment, but this is what we're talking about today, what I'm talking about today. Are there any questions so far? No? Okay. Then I'm throwing up a slide that I made recently for a book chapter. And this is just to kind of give kind of a high-level overview over different types of models. And I have organized them here kind of in terms of their level of abstraction, so less abstract and more abstract, and in terms of what level of the biological organism are we modeling, so that there are subcellular models that look at the molecular networks inside the cell. There are cellular and synaptic models. Then we can go to circuits, entire systems, or the organism. And there are some terms here that we have already seen. So there's integrated fire here. There's Hodgkin-Huxley type. The Blue Brain Project I placed kind of here, and the Human Brain Project up here. You don't need to be familiar with all of these terms, but the point is again that while I'm talking today just about conductance-based single neuron models, modeling happens at all kinds of levels and different abstraction levels in neuroscience. So there is a rich playing field out there to explore.
Just a question, like you put a small wall in circuit level. I mean, usually I would say usually I read that more referring to a more system level, as in.
Yeah. I think all of these can be shifted around depending on where exactly you place them. Yeah. Small networks, I agree, can be systems level, can also be circuit level. Yeah.
You would read it as like the internet
Right.
has to be a small wall.
Right. And I encourage you, as you become you are a computational neuroscientist, to make your own version. You know, it's like, why did Astrid put that over here? I think it really belongs over here or something. So again, this brings me to this point again, and I'm going to sound like a broken record. But as you look at the large number of possibilities that you have here, the way to kind of make sense of this forest for the trees, I don't know if I said that right, is to keep in mind, what am I actually asking? What scientific question do I want to answer? And of course, if you're interested in dendritic computation, you cannot do an integrated fire model. If you're interested in systems level, you might want to use integrated fire neurons so that at the neuronal level you're simple, but then you put them together in complex networks. So always keep that in mind. And what I'm going to talk about today is really at the cellular level. I actually even took out the multi-compartment part that I had. So we're really sticking with single compartment models and Hodgkin-Huxley type models here. Okay, so conductance-based models. These next couple slides we can go through relatively quickly because of Tim's talk. So we have ion channels. There are different types. You already are now very familiar with the sodium type. There are, as Tim emphasized, different potassium types. So actually the family of potassium channels is, as he pointed out, much richer than the sodium channels. And I had not thought of that angle of evolutionarily it's safer to play with a negative feedback than a positive feedback. You might break a positive feedback in many different ways. So I think that's reflected in the fact that there's such big diversity of potassium channels. Calcium channels can also be important, and then you have pumps that maintain or restore these ion concentration levels. And one model that I'm going to talk a little bit about today before I move into the fly model is a stomatogastric, again, the same stomatogastric model neuron that I talked about before with the ×8. So here's now a schematic of the different types of membrane currents that are present in these crab stomatogastric neurons. They have all been characterized experimentally in terms of voltage clamp. And you see that now we now have already 8 different membrane currents. And I think there might still be some in there that haven't been found yet. So there's really, again, this rich dynamical potential. So in this particular model, we have your typical fast sodium current. We have 2 different types, a fast and a slow calcium current. Several different types of potassium channels with different properties. Who has heard of the H current? Some of you have. So this is kind of a freaky current in that everybody else, like in Tim's Hodgkin-Huxley diagrams of their activation curves as a function of voltage, everybody else activates for depolarization. This one activates for hyperpolarization. And that actually has some very interesting consequences for the dynamics of this neuron. And it's not so exotic that it just exists here. Actually, many, I want to say maybe even most neurons have an H current, and so that is a very interesting thing to think about. What does that add? Another feature that it has, which is also interesting, is it's extremely slow. So it's basically, in terms of its dynamics, the opposite of the sodium channel. In this particular type of neuron, the sodium channel activation is on the order of a millisecond or less. The H current activation is on the order of several seconds. So that adds a whole other dimension to what these neurons can do. Okay, so then again, this can be fairly quick. As a reminder, some channels, like the voltage-gated sodium channel, have not only an activation gate, and mine looks a little different from Tim's. I think it's blue and it's horizontal, but yours was other color and vertical. And we have a little inactivation gate. And so then the dynamics during the action potential is that first your activation gate will open up. Sodium can flow down its gradient into the cell. That makes your upstroke of the action potential. And then later, because this is a slower gate than this one, the inactivation gate slams shut. And these gates are now, again, this is just a reminder, described by these variables M and H. And the reason I'm emphasizing it again is that one thing I want to achieve with this talk, and it worked a couple of weeks ago when I taught at a course with almost purely molecular biologists, so I hope that you guys should get it easily, is the translation of these biological entities into math and how then that shows up in the code. So we'll look at the code together and you'll hopefully recognize a lot of lines in that code, and you can then say, aha, that's the M gate, that's the H gate, and so on, to kind of give you a sense of what's happening where in the code. So this activation and inactivation is playing into this equation. And I've now grayed out the calcium dynamics part because I'm not talking about that today. And again, as a reminder, we have in this model these 8 different membrane currents, and here you see their activation and inactivation curves. And you see that except for the H current, they all have this typical sigmoidal shape with high activation probability at depolarized membrane potentials, but you see that they differ, and especially the inactivation gates differ not only in terms of at what action potential, at what membrane potential they are at half value, for example, but also in terms of their steepness. So there is this commonality between them all, but they also have subtly different features, and that's again what plays into the rich dynamics. And so those channels are then described by these. So these are the actual mathematical expressions for the activation curves and inactivation curves. And then you have this whole additional dynamic potential in terms of the time constants, which channels will activate more quickly or more slowly when there's a voltage change. And I wanted to point out here, this is a logarithmic scale of time constant, right? So you really have some things happening at a sub-millisecond level and then up to several seconds. Okay, so how does all of this play together? Oh, finally, so these are then the equations that describe these time constants and how they depend on voltage. And when I first started learning about these conductance-based models and about Hodgkin-Huxley, I always found it confusing that they're called time constants when they're really dependent on voltage, but that's just the historical terminology. Okay, and then together these activation curves and the time constants play into these differential equations. So what does all of this, I keep harping on this potential for rich dynamics, what does all of that look like? So this is from earlier work that I did where I did one of these parameter space explorations, namely taking this stomatogastric model and varying some of its parameters. So this thing, at some point I counted, and if you look at all of these places where you have parameters, right, you have a half activation voltage here, you have this factor here that plays into the time constant, this factor. So there's tons of parameters in here, and I think this particular model has about 140 parameters. So that is a huge parameter space, and even though computer power is constantly increasing, I had to seriously cut down what parameters I'm going to vary in this space. It was just computationally not feasible to explore 140-something dimensional space. So what I decided to do is to narrow it to just 8 parameters, and these are the maximal conductances of these different ionic currents, right? So you have, what am I going to show here? Yeah, so you have these 8 different currents, and each of them has what's called a maximal conductance. Tim, I don't think, used that term so much before, so I have to make sure that everybody follows what this is. If you look at the equation, the current is a conductance times a bunch of gating variables times a driving force, and that's basically a version of Ohm's law. And if you now think about what would happen if all the channels were fully activated, so M equals 1, and fully de-inactivated, these are complex terms, so this would also be 1, then that would mean all the ion channels of that type would be open, and that's when your membrane conductance for that ion type is G bar. So this is if all the channels of that type are open. They almost never will be because these factors are between 0 and 1, but that's what a maximal conductance is. So it's basically a measure for how many ion channels of that type are present in the membrane. If you have millions, this will be a large maximal conductance. If you have, hundreds of thousands, it will be smaller. So that's the parameters I varied. So I built this database where I have 8 dimensions. Each dimension is a maximal conductance. Sarah is nodding because she knows all of this. So this is an 8-dimensional space, and I covered this with, in that case, it was 1.7 million different points on a grid. So each of these parameters could take 6 different values. Now I'm launching into my work that I was really only going to touch on, but maybe this afternoon. So covering all of this with a grid, and then you can just ask for every parameter combination on that grid. Of course, this is all automated. What is the behavior of the model? And I'm showing you some examples here. So what we're looking at here is voltage traces for a bunch of different versions of this model. And over here, these bars show you how big is the maximal conductance for these different membrane currents. So this one has a lot of transient calcium current, but not so much delayed rectifier current, and so on. And you see that leaving everything else, all of these over 100 parameters constant, just by changing the mixture of ion channels in that membrane, you can get extremely different behaviors out of the same model neuron. So some are silent, some are spiking, but it turns out this one is spiking, but without a sodium current. So you can actually have a fast calcium current take over the role of a sodium current in generating what's then called a calcium spike. This is a more typical spiking neuron. And what we were particularly interested in, because we were studying these bursting neurons, these stomatogastric neurons are bursters. They're also most of them motor neurons and part of a central pattern generating circuit. So we were primarily interested in bursting, and there are many different versions of bursting that can come out of this model. So that was my introduction to conductance-based models. And now I'll launch into this particular model that you'll be playing with if you feel like doing exercises this afternoon, and that is a single compartment model of a fly larval crawl motor neuron. So this is primarily in collaboration with Stefan Pulver at St. Andrews University, and he likes to call them maggots. So these are the little fly larvae that crawl around in the dish, and they have a central pattern generator that does that, and these are some of the motor neurons involved in that. And as I mentioned earlier, we were interested in them partially because of that involvement in the epilepsy mutant, where a messed up sodium channel actually, led to an epilepsy type behavior. And I have to say, since we talked about funding earlier, the postdoc Cengiz Günay, who is now faculty at a local Atlanta area college, he actually managed to get from the American Epilepsy Foundation a postdoctoral fellowship for that. And so a lot of people had told us, no way are they going to fund fly research in the context of epilepsy. So in that case, they agreed with us that this is a good model system to kind of understand at a basic level how can seizures arise. Okay, so I'll say a few words about this model, and then we can go into the lunch break and pick it up from there. This is almost the end of my lecture anyway. So let me first tell you what is this model, and then I'll give you a preview about what we'll do this afternoon if you're interested in doing some exercises. Okay, so this is a spiking fly larval motor neuron model. Over here is an output of that model, and this looks actually very similar to the output that you get from one of these actual neurons if you record from them and you inject a little bit of current starting here. And I have a file that I gave to Simone for posting that has a link to a paper if you're interested more to see some experimental traces of these neurons and interested in the background of all of this. But for the purposes of our exercises this afternoon, this is a relatively simple conductance-based Hodgkin-Huxley type model neuron. It has a relatively minimal set of ion channels, so we know actually that there are some calcium channels in these neurons that we haven't even included in the model yet. So for now, it just has a leak current, two different types of potassium currents, a transient sodium current. This one's fairly standard with the inactivation gate here. And then some of the exercises will explore the role of this persistent sodium current. So that, as you see, doesn't have a gate, so it's a non-inactivating or persistent channel. So if you make a voltage step, this channel will open up because of its activation gate and then will close down because of its inactivation gate, whereas this channel will just stay open as long as you maintain that voltage step. Okay, and so it's based on this fly motor neuron. And so what will we do with this? The intent of these exercises is that if you're interested in this kind of model, that you just get a feel for what does the actual code look like, what can you do with these models, and just play around on a couple simple exercises. And so what will that look like? After lunch, I will show you to use how to use this XPP system, which is really a differential equation solver made specifically for these neural type simulations by Bart Ermentrout, a mathematician in Pittsburgh. So you will learn how to run simulations with this, how to change model parameters, and if you're interested, work on exercises. Now, yesterday, because I realized that I didn't have it running on my computer, I had to do the installation yesterday from scratch. And it is fairly simple, and that same file that has the reference to the paper also has a reference to a specific website where you can find and download and install XPP. It has a Mac version and a PC version. I happen to, for historical reasons, be a PC person. But if you want to use it, you can either look at the details that are online. The instructions that are online are fairly detailed, so you might be able to get the Mac version running, or maybe you could work with someone who has a PC. How many PCs do we have here? Oh, yeah, okay, that's maybe again about half. So,
We take that into account when selecting.
is there a penalty or a reward for having a PC? So this is the, so because the installation has to happen before you can use this, it took me only about 5 minutes yesterday, but there can be some technical glitches. So if you're motivated, I think we're reconvening at 3, right?
We will reconvene at 2:30.
2:30. If you're motivated and you have some time after lunch, you could already start looking at this file and seeing if you can get it to install on your computer. So I don't know where Simone put it now. I gave it to her earlier. So she's still establishing a kind of a website.
It will be in the Dropbox folder that is or will be shared with everyone.
Right.
Don't share with me, I just don't know whether it's shared with everyone.
And if you can't,
But there is a central place where all these things go,
Right.
and where you will find slides and all that stuff will be shared as soon as we get it from time.
right. And if you can't, if you don't have time or can't get it to run by 2:30, then those of you who even want to do these exercises, we can just try to figure it out together. These exercises are voluntary, right? Except for the exam in the end. Exercises are voluntary, right?
They're encouraged, but there's no exam or test, or
So can I just
there's no penalty or punishment.
Can I just get a ballpark raise of hands? How many people would like to play around with this thing? Oh, excellent. Okay, so then we'll reconvene at 2:30 with or without your installation working.
So would it be useful if they have it running for your second part of the lecture?
Well, there will actually be only a few more slides in the lecture, and then I was planning to open it up here myself and show how it's being used.
But then they should
So-
install it.
But should people have it installed at that point, or that doesn't matter?
Right, so we will reconvene at 2:30.
well, there's-
2:45. So I think it makes more sense for people to have it installed than if you talk about it, I think.
It does, but you might get stuck because there's always something, someone's computer, depending on operating system. The important thing is, at least on PCs, that you carefully follow the instructions, and I have those. They are on Bart Ehrmantraut's website, so this is the general homepage. No, that's not showing. I have to get out of this, I think. Why doesn't this show? Okay, let's do escape. Okay, now we should be able to see it. Okay, so this is the website. That link is also in that magical file that hopefully Simone has made available. And in particular, there is an easy window install. And you see it's relatively short. The important thing is that you really put it into this root directory. Otherwise, paths will get messed up and links will get messed up. Okay. On the spot, 1 o'clock.